Welcome. In this session, firstly, I'm going to discuss about the remaining methods of sex determination. Then after which I'll also discuss about mutations, and I'll also start discussing about pedigree analysis. So let's begin this session with sex determination. So in the last session, I discussed two methods under this. One is a XYXX method of sex determination, and another one is a XOXX method of sex determination. In both those methods, we have seen male heterogamity, which means males are producing two types of gametes. But here I'm going to discuss another situation where the males will not be producing two different types of gametes. Instead, the females will be producing two different types of gametes. So that will be ZZ ZW method of sex determination. So that is ZW method of sex determination. So in this case, the females are heterogametic. The females are heterogametic and they will be having one Z chromosome and one W chromosome. So that is a Z chromosome and a W chromosome, okay, are present in case of females. Coming to males, so males are homogametic. So these will be having, okay, two Z chromosomes. So they have two Z chromosomes. So they have two Z chromosomes. Okay. So here, this method of sex determination we will be seeing. Okay, in case example here, in case of birds, okay, some fishes, then insects like butterflies, okay, etc. In them we may find such type of sex determination, right? So females here are heterogametic and males are homogametic. So let's see how, okay, the method, I mean how the sex is determined in these uh, birds or butterflies. Here AA plus, okay, ZZ. So these are males, right? And coming to the females, we see here AA plus ZW. So these will be parents 2N. Okay, right? And now during the formation of gametes, we see here the sperms will be always having Z chromosome. So that is 100% of sperms. So that is the reason why I mentioned that males are homogametic. And coming to females, they are going to produce two types of gametes, one with Z chromosome and one type with the W chromosome. So that is 50% of X will have Z chromosome and 50% of other X will be having W chromosome. Right now, the egg, when comes in contact with the sperm having the Z chromosome, it will be AA plus ZZ. So this will develop into a male individual. And when the sperm with the Z chromosome comes in contact with the egg with the W chromosome, it will be AA plus ZW, and this will be the female individual develops. Okay, so this is the ZW method of sex determination. So in this case, as the females are heterogamity, so here the condition, we call it female heterogamity. So female heterogamity is seen. Okay, so I told you already in earlier case XY, XX and XO, XX we have seen male heterogamity, and in this case we will see female heterogamity. Okay, so that's all about ZZZW method of sex determination. So now we will see another method of sex determination in honeybees. So let's see sex determination in honeybees. Okay, so here in honeybees, this sex determination is also called haplodiploid. Okay, sex determination. It's called haplodiploid sex determination. Right. So why it's called haplodiploid sex determination? In case of adult male honeybees. So here the adult male honeybees are haploid. So they are haploid. Okay. Whereas the adult female honeybees. So the adult female honeybees will be diploid. Right. So I'll just mention here the diploid number of chromosomes. The diploid number of chromosomes in honeybees is 32. So they have 32 chromosomes in the diploid condition. So hence the adult female honeybees will be having 32 chromosomes, but the adult male honeybees who are haploid will have only 16 chromosomes. Okay, so let's see the cross between a male and a female honeybee. Okay, so male honeybee. So I told you male honeybee is having only 16 chromosomes, and the female honeybee is having 32 chromosomes. So parents to N. Right. Now here, as the honeybee, that is male honeybee, is haploid. So it's already haploid. So hence no meiosis takes place in its reproductive organs. Instead, mitosis will take place. Okay. So that's, I'll just mention mitosis. So mitosis takes place in its reproductive organs, and it results in the formation of sperms with 16 chromosomes. So these are sperms with 16 chromosomes. Now coming to the female honeybee, it is diploid. The female honeybee is diploid. I'll just mention here N, and in this case 2N. So it's already, it's diploid. Because of this, it need to have meiosis in the reproductive organs. So in its reproductive organs, meiosis takes place, and it results in the production of eggs with the 16 chromosomes. So these are eggs with the 16 chromosomes. Now, when the eggs are fertilized by the sperm with the 16, okay, then of course 16 plus 16, it results in the production of 32, so which is diploid, and this will develop into female. So will develop into female. Okay, so here the adult honeybees are called drones, and in case of adult female honeybees, we have here queen and also the worker bees. So queen and worker bees, both of them will be in diploid condition. It is only the males who are called drones will be okay, haploid. So here the egg, okay, with the fertilized with the sperm, so this will be with fertilization, okay. So in this condition it's a with fertilization. So now if without fertilization, so here egg, so these eggs without fertilization, so if no fertilization takes place, okay, of course without fertilization also the egg is capable of developing into a new individual, and this will develop into a male. So this will develop into male. So it means that the males develop without fertilization, whereas females will develop with fertilization. And this is the reason, okay, in case of males, okay, male honey bees do not have father. The male honeybees do not have father but have grandfather. So the male honeybees don't have father but will have grandfather. Okay, because you can see here the eggs are developing without fertilization. So hence there is no father for this male. But the mother, okay, so mother is developed okay, from the fertilization of the egg. So the mother will have father. Okay, so it means this male will have grandfather but not father. Right, so this is the case in case of sex determination in honeybees. Right, so that's all about sex determination. So the next concept which I would like to discuss is mutation. So the next one we'll see about mutation. So what is mutation? So mutation is, okay, simply alteration of DNA sequences. So mutation is alteration of DNA sequences. So here if you ask me a question that what a gene is made up of, so my answer will be it is just composed of a sequence of nitrogen bases. So each gene will have its own particular sequence of nitrogen bases. Okay, say suppose here we have some gene X. So this gene X have some sequences like AGTA something. So this is how you will see, okay, the sequence. Of course an average gene will have 3000 nitrogen bases. So here just for example I have taken to make you to understand that what actually is mutation. So say suppose this is the entire sequence of a gene X, right? And now during the process of copying, what happens sometimes, okay, some of these nitrogen bases will change. Okay, so they may change. So here if you are considering here guanine, so this guanine may have changed into adenine or into thymine or other nitrogen base. Okay, and in some cases these nitrogen bases may be deleted or they may be added. Here let's say this one, okay, so here the guanine is changed to, okay, T is changed to T. So here A is adenine, G is guanine, C is cytosine, and T is thymine. So these are the nitrogen bases that we see in case of DNA molecule. And here, okay, the G is replaced with the T. So the G is replaced with T. So once it is replaced with T, then the sequence will be A G C T A T C C A T A G. So you can see there is a change in the sequence of nitrogen bases. So that's what I say, the alteration of DNA sequences. So now this is a mutation. Okay, so here the G is replaced. So here you can see this G is replaced. Okay, and this is what I call it, a mutated gene. Is called a mutated gene. Okay, now I'll classify these mutations into two types. One I call it point mutations, and another one called frame shift mutations. So point mutations and frame shift mutations. So here, in case of point mutations, there is a change in a single base pair. There is a change in a single base pair. So in short, I'll write small b, small p. Okay, those things will understand in our coming sessions what actually is this base pair and all, when I discuss DNA structure in detail in the next chapter, molecular basis of inheritance. Okay, so if there is a change, right, in a single base pair, then the condition is called point mutations. Okay, so if there is a change in single base pair, okay, then in this case the frame shift mutations. So if there is, okay, addition or deletion of base pairs, then the condition is called frame shift mutations. Of course, here the base pairs of DNA. So change in a single base pair of DNA and addition or deletion of base pairs of DNA. Here, which I have explained in the first instance, where I told that one nitrogen base is replaced with another. So this is actually an example for point mutation. So this is a point mutation. Okay, so already I mentioned in point mutation a single base pair is changed. So you can see here, right, the G is a base, nitrogen base, replaced with another nitrogen base. So such mutations, we call them point mutations. Now let's see, in case of frame shifter mutations, actually the gene will be read from one direction to another direction. Okay, so at present I cannot explain you all that, you will slowly understand in coming sessions, right? So here just I tell you that the gene will be read in a particular direction, and usually we will be reading in three nitrogen bases. So here, okay, 3, okay, and the next 3 and the next 3 and the next 3. So this is, and say this is the direction of reading the mutation. Okay, now coming to point mutation. So I'm going to come to that point. Right, so I mean coming to the frame shift mutations. Frame shift mutation. So in case of frame shift mutation, right, we'll take a sequence A, G, C, C, T, A, some sequence I have taken, and I told you, okay, again we'll say this is some gene Y, is a gene Y, and I told you that the genes will be read in a particular direction, and say this is the direction, and it will be read, okay, three letters. Okay, so this is how we see, right? And okay, now say if there is an addition, so somewhere here, okay, between the C and C there is an addition of a nitrogen base A. So addition of nitrogen base A. So if it is the case, then the reading frame will change from that point of, okay, addition. So AGC, CCA, TAG, C, now here ACC, ATA. Now you can see, so this one remains as it is, and here, but here we can see instead of CCC we have a difference, okay, CAC, and then next CAT has come. Actually it should be ATA, but it has come with CAT, and so on. So there is a change in reading, okay, sequence from the point of addition. Okay, so from that point of addition there will be a change in the reading frame. So what does these three letters actually code for? Actually these three nitrogen bases will code for one amino acid. So that I'm going to discuss in protein synthesis in the next chapter, molecular basis of inheritance. Okay, so here just I'm going to tell you briefly what is mutation. So here the addition of nitrogen base has changed that reading frame. Okay, so that's what there will be a shift in the frame. Okay, so that's we call frame shift mutations. Now I told you that there will be addition, or in other cases there may be deletion also. So if there is a deletion, so let's see. So in this case I'll take the same one. So here, say this or I say here this A is deleted. So this nitrogen base is deleted. Then, so how the sequence will be AGC, CC, the next one will be TAG, CC, ATA. Now, right from that point, we'll see a change. So AGC will remain same, that now it has become CCT. Earlier it was CCA. The next one it is TAG. Now it became AGC, then CCA. You can see right from that point the reading frame is changed. Okay, so there is a change in reading frame. Okay, so that's what the frame shift mutations. Okay, so that will actually result in a production of different right polypeptide, different polypeptide. So earlier this may produce a functional enzyme, and in this case it may produce a non-functional or no enzyme at all. So I told you in the beginning, so if the mutated gene have several possibilities. So it may produce a functional gene, it may not produce the functional gene, or it may, okay, not at all produce the enzyme. Okay, I mean any polypeptide chain. Usually when a gene is expressed, that will be in the form of a polypeptide chain. So that we will understand slowly in our coming sessions. Okay, so that's about two types of mutations: the point mutations and frame shift mutations. Now we know here the chromosome. So here chromosome, what actually is this chromosome is a long DNA molecule that is coiled upon coil, that what we call supercoiling. So there is a lot of coiling takes place in case of DNA molecule, and that results, just I'm mentioning here coiling. Okay, so it's a coil, so there is a coiling of DNA, and this is only one DNA molecule that will be seen from one end to the other end. So this is, we will be usually drawing the outline of this chromosome. Okay, so this is the centromere, and okay, so this is a chromosome. So what does a chromosome made up of? Chromosome consists of DNA, a long DNA that is, okay, super coiled and seen from one end to and another end. So there is only one DNA molecule. Okay, so this is DNA molecule that is supercoiled. So DNA molecule that is supercoiled, and it extends from one end of the from one end of the chromosome to the other end of the chromosome. Okay, right. Now if there is a change in the DNA molecule, so just now we have seen a point mutation. Okay, so any change in the DNA molecule will definitely have effect on change in the chromosome also, because the chromosome is nothing but the DNA molecule. Okay, right. So that's, I'll write, so here if there is, okay, alteration, or I'll say, so if there is a addition, so if there is addition, okay, deletion or duplication, okay, of DNA segment. So if there is addition, deletion, duplication of DNA segment, okay, this results in, okay, alteration of chromosomes also. So you can see that there is an alteration of DNA. If there is an alteration of DNA, then we will also see the same alteration in the chromosome also, because I told you the chromosome, okay, is made up of DNA molecule. So a chromosome is nothing but a highly coiled DNA molecule that extends from one point of the chromosome to the other point. Okay, and we know that the genes are present on chromosomes. Okay, and alteration in chromosomes, so alteration in chromosomes results in, okay, results in abnormalities, okay, or aberrations, right? Abnormalities or aberrations in the individual. So that part I will be discussing at the end, okay, especially the chromosomal disorders. Okay, so alternation in chromosomes will result in abnormalities or aberrations. Now here there are certain agents that cause mutations. Okay, so here those substances that cause mutations, so substances that cause mutations are called mutagens. Okay, substances that cause mutations are called mutagens, and these mutagens can be physical, okay, mutagens, then They can be chemical mutagens and also biological mutagens. So here physical mutagens, like in case of UV radiation, okay, chemical like example DDT, and in case of biological viruses. Okay, right. So that's about mutagens, and again this topic mutation I'm going to discuss in the chapter molecular basis of inheritance after I discuss genetic code, right? So that's all about the mutations. Then the next concept I'll see is about pedigree analysis. So I'll see. So next one I'll see about pedigree analysis, right? So first we'll say about the definition. So here the definition is, it is analysis, okay, of, right, several traits. So it will be analysis of okay, I mean traits of several of generations of a family. Okay, so it is analysis of traits. Okay, so that is analysis of traits in several of generations of a family. The next one I'll say about pedigree analysis. So before I discuss in detail about this pedigree analysis, first we'll see about the definition. Okay, it is analysis of traits in several of generations of a family is what we call pedigree analysis. So it is analysis of traits in several of generations of a family is called pedigree analysis. So here we are going to use this pedigree analysis to study the inheritance of traits in human beings. Okay, so here it is helpful in studying inheritance of traits in human beings. Okay, so here in case of human beings, we cannot make a controlled process that how we have performed in case of a pea plant, okay, in other animals, right? That is not possible in case of human beings. So hence we need to use this method, that is pedigree analysis, to see a particular character, how it is being inherited from, okay, one generation to another generation. Okay, right, and we call that as a pedigree chart. And to draw a pedigree chart, we need to use certain symbols. Okay, so first we'll see those symbols. So symbols used in drawing pedigree charts. Okay, so symbols used in drawing pedigree charts. Okay, after coming to know the symbols, then I will show you how to draw a pedigree chart. And I told you the pedigree analysis is useful in studying inheritance of a particular trait. So how it is being inherited, okay, whether it is dominant, recessive, whether it is present on autosome or it is present on allosome, all these we can come to know, okay, by drawing these pedigree charts. Okay, so we'll see first the symbols that are used in drawing the pedigree charts. So if I'm using a symbol, okay, then this one, it's a male, and this symbol indicates female. And if I'm drawing in this way, so this indicates sex unspecified, which means neither male nor female, right? And if I'm going to shade these symbols, okay, so I'm shading these symbols, right? And so in this case, it indicates that these are the individuals who have the trait. Okay, so that's what I mentioned here, affected individuals. Affected individuals. Okay, so whenever we see, okay, the trait in that, okay, in the sex unspecified one, or in the female or in the male, okay, we need to shade that. Then it indicates that person is, okay, affected or carrying that particular trait. Now if I'm going to put a line between the male and female, so in this way, this indicates marriage between them. Okay, and if I'm putting two, okay, lines, then it indicates it is a, right, consanguineous marriage. So it indicates a consanguineous marriage. So consanguineous marriage, which means this is a marriage between, so marriage between relatives. So marriage between relatives. So those marriages among the relatives, okay, what we call consanguineous marriage. Then if I'm drawing, okay, something like this, just mention here. Okay, so here a line below this and indicating here. So this indicates the above one are parents and the below one are offspring or progeny. Okay, so parents and below are offspring or progeny, right? And here we need to draw in the order of birth. Okay, so here the left to right will be the order of birth. So here the first born should be drawn on the left side. So this is the first born and this is the second born, and if the third one comes, it will come on the right side. Okay, okay, so all those new borns will be on the right side. So the left one will be the, okay, the elder one, and the right one will be the younger ones. Okay, so this is the order of birth that we need to mention. So whenever we draw in this way, it indicates that, okay, in that family the first one born is a daughter, then later they had a son. Okay, so this is in the order of birth. Then if I indicate in this way, okay, it indicates that they have a son and the son is affected, or he is the one carrying the trait. So here it indicates both the parents are normal but their son is affected. Okay, and if I'm, okay, putting, if I'm going to put a number here in this way, then it indicates 5 unaffected progeny. or offspring. Right. Okay, so that indicates 5 unaffected progeny or offspring. So of course there are many other symbols that we use while drawing a pedigree chart. Those I will be discussing when I take the neat part of this chapter. Okay, so first we'll see by using these symbols how we can draw a pedigree chart. So here we'll see an example. Okay, inheritance of autosomal dominant trait. So we'll see inheritance of autosomal dominant trait. So here autosomal dominant trait, which means okay, the trait is located on autosome. So I told you that except the sex chromosomes like X and Y, the remaining all other chromosomes are called autosomes. Okay, so here in this case, when I say autosomal dominant trait, which means that trait is located on okay autosome and is dominant and is dominant. So here let us understand this by taking an example. Okay, myotonic dystrophy. So myotonic dystrophy, this is a genetic disorder that later we will see. So myotonic dystrophy, and this is a autosomal dominant trait. So myotonic dystrophy is a autosomal dominant trait. It's an autosomal dominant trait. So let's see in this case. So here a female who is having this myotonic dystrophy. So that's why I shaded this. Okay, so earlier in the symbols, and I mentioned, I told the person carrying the trait should be okay shaded, and here is a female who have the problem myotonic dystrophy or carrying that trait myotonic dystrophy, marries a normal man, now they have 4 children. So they have 4 children, and the first one here is a daughter. So the first daughter is normal. Second, they had a son. So son inherited this trait. So son inherited okay that trait. And next they have another son. So this is the third okay one, and this one is normal. And lastly they also have one more daughter, and daughter also inherited the trait. So inherited the trait. So here I'll just mention the order. Okay, so this the eldest one in the family, first one, the second, third, fourth in the birth order from left to right. Okay, right. So total 4 children, so 2 sons and okay 2 daughters in this family. Now so here the daughter marries, say suppose again a normal man, marries a normal man, and now they have okay 3 children, say okay they have 3 children. So first one is son, and second one is a daughter, and third one is again son, right? The daughter inherited this trait. So this is the first, second and third one. Okay, now the normal male marries a normal woman, and now they have, okay, children. Of course, this is what I'll mention parents here. Then, okay, so these are all the F1 generation. So this is F1 generation, and these will be the F2 generation. Okay, and now here, coming back to this, the normal male married a normal woman, and they have three children. Okay, so first one is daughter, second one is also daughter, and third one is a son. Now in this case, okay, all the three are normal, so none of them, because none of their parents have this. Okay, so this is the order 1, okay, 2 and 3. Right, so now here we can see that every generation there is, okay, the affected individual. So indicates that in case of autosomal dominant traits, the pedigree, okay, the pedigree chart is vertical. So which means we will see the character or the trait in every of the generation, and the transmission of trait from parents to either of the sex, we see it. So transmission of traits, okay, from parents to either of the sex is seen. Okay, so of course in detail you will be studying in your practical session. So in practicals you have this pedigree analysis. There you will come across many other examples and many other conditions. Okay, so I told you the pedigree analysis will let us know that whether the trait is autosomal or allosomal, dominant or recessive conditions. Okay, right. So in case of this autosomal dominant, we will be seeing that the trait from the parents, okay, it will be transmitted to either of the sex, and the pedigree chart will be seen, okay, vertical, and the males and females are equally affected in case of this autosomal dominant trait. So if we see such conditions, we can come to know, we can come to the conclusion that the trait is autosomal dominant. Okay, so that is about the inheritance of autosomal dominant trait. So we'll also see how the inheritance of autosomal recessive trait takes place, and that part I will be discussing in the next session. Okay, so this is the pedigree chart in case of autosomal dominant trait. So this is the pedigree chart in case of autosomal dominant trait.